Okay, so good afternoon. So today is the 10th problem solving session for the course Environmental Chemistry and Microbiology. The course instructors are Professor Anjali Pal and Professor Sudha Goyal from IIT Kharagpur. And today we will be discussing questions from microbial growth, questions related to the topics of microbial growth kinetics, all the topics that have been covered in the 10th week lectures. Okay. So starting with the first question, question number 1. The process in which all living cells, spores and viruses are completely destroyed from an object is called. So option A is disinfection, option B is pasteurization, option C is sterilization, and option D is antisepsis. So which of the following process defines which of the following terms is actually used to define the phenomena where all living cells, spores and viruses are completely destroyed from an object? So do you know what will be the answer?
See, ma'am.
Yes. So the correct answer for this question is sterilization. So sterilization is the process where all the microbes are completely destroyed. So for the definition, it will be sterilization may be defined as the complete destruction of all the microorganisms, including the most resistant bacteria and spores. Sterilization is achieved at high temperature and high pressure by autoclaving. So when we use the term sterilization, that means we are destroying all the microorganisms, whatever like fungal spores, bacterial cells, any other viruses, all types of microorganisms are destroyed. So generally sterilization is done for some abiotic material or abiotic surfaces like non-living surfaces or non-living materials. So let's say I have a medium that I need to sterilize. When I say that, I mean I have a medium from which I want to kill all the microorganisms and even their spores. Now spores are very resistant structures. So bacteria makes the spores so that it can survive at harsh conditions. So we have to do the sterilization such that spores are also destroyed. So we have to use temperatures at which the spores also cannot survive. So that's why it is generally done at high temperature and high pressure. Autoclaving is a process by which we can achieve these high temperature and high pressure. So I'll explain autoclaving in the subsequent slides. So the other options in the questions were disinfection, antisepsis and pasteurization. So let's see what are those. So disinfection is the destruction of vegetative pathogens by using physical or chemical methods such as chlorination or ozonation. So what is the difference between sterilization and disinfection here? So in sterilization we can see it is destruction of all the microorganisms, whereas in disinfection we are only destructing the vegetative pathogens, the pathogens that can divide, that are continuously dividing, that are in vegetative state. We can destroy those pathogens, and this disinfection is generally done by using some physical or chemical methods like chlorination, ozonation. So you must have seen people adding some disinfectant solutions in the water to mop their floor surfaces at home, right? So the Lysol solution or the Dettol solution, those are mainly disinfectant solutions that can kill the pathogens that are present on the floor surfaces, or using a sanitizer for sanitizing your hands. So that's a process of disinfecting your hand surface. Okay. The next term is antisepsis. So, antisepsis is the destruction or inhibition of microorganisms on living tissue to prevent their growth by using antimicrobial agents. Now, both sterilization and antisepsis is the destruction of microorganisms. In antisepsis, the another term that is used is inhibition. So, during antisepsis, the microorganisms are not always destroyed, they are sometimes inhibited. They can be inhibited also. Inhibited means their growth is uh, like the microorganisms are not killed, but the growth is stopped. Okay, so they cannot grow further. So that is the process of antisepsis, and it is done on living tissue. So whenever a person gets a wound or any kind of physical injury, you have seen people putting the Dettol solution to wipe the wound. So that is a part of antisepsis. That's an antimicrobial, like it has antimicrobial properties, so it can prevent the growth of any microorganisms on that tissue. Or during any surgery, the patients are generally prescribed antibiotics. So antibiotics are also antimicrobial agent that can inhibit the growth of certain bacteria at the affected living tissue. Okay, so antisepsis is the term used for destroying the microorganisms on living tissue, whereas sterilization is the term used for destroying the microorganisms on some object or abiotic surfaces. Okay, pasteurization is the last term. So pasteurization is a process of partial sterilization where only pathogenic microbes are killed. So pasteurization, it's also a type of sterilization, but it is a partial sterilization. So in sterilization we are destroying all the microorganisms that are present, whereas during pasteurization we are only destroying the pathogenic microbes. Okay, so you must have seen this term on written on the milk packets. Generally milk that are available in the packaged forms, those are generally sterilized in the industries using the process of pasteurization, where it is heated up till a certain temperature and then rapidly cooled down to destroy any pathogenic microbes that are present in the milk. Okay, I hope all these 4 points are clear.
Yes, ma'am.
So we'll move to the next question. Question number 2. What is the generation time? So out of the 4 following definitions, you have to say, you have to tell which is the correct definition for generation time. Option A is time required for microbial population to generate energy in a culture. Option B is time required for a microbial population to double in number. Option C is time required for a microbial population to double in weight. And option D is time required for a microbial population to reach stationary phase. So which of the following definition is defining generation time? Can you answer? So generation time, the correct answer is option B. It is the time required for a microbial population to double in number. So now you see the term generation time. That means the time taken to make a new generation. So what happens in bacterial cells? They divide by process called binary fission. So you can see in this process you have one bacterial cell. So you have one bacterial cell, and then this one bacterial cell, first the genetic material multiplies into 2, and then the complete cell divides into 2 daughter cells. So one cell divides into 2 daughter cells, and then in the next generation this cell will divide into 2 cells and this cell will divide into 2 cells, giving total 4 cells. So at each generation the cells are dividing to form two cells. So that means the cells are doubling in number. Okay. So generation time is the time it takes for a population of bacteria to double in number. That is how we define the generation time for bacteria, because it is the time that is taken by the bacteria to make a new generation. What do you mean by making a new generation in terms of bacteria? Bacteria, after each generation, they are doubling in number. So it is the time taken for a population of bacteria to double in number. Okay. So for many bacteria, the generation time ranges from minutes to hours. So it can be like few minutes, like 20 minutes for a particular bacteria, and it can be up to 1 or 2 hours for some other type of bacteria. So it depends from different species and different genus of bacteria. The generation time differs. So some bacteria can divide very rapidly, and some bacteria are dividing very slowly. Okay. Because of binary fission, bacteria increase their number exponentially, whereby their population doubles every generation time. So because of this binary fission, what is happening? They are increasing in number exponentially. So you see, if I ask you, what will be the number of what will be the number of bacterial cells in a population after 3 generations? So how will you define? You will define by this term, p is equal to 2 raised to the power of n, where n is the number of generations. So let's say after one generation, the number of cells, like if we start from one cell, the number of cell is becoming 2, because 2 raised to the power of n, number of generation is 1. After second generation, what will happen? N will be 2, so 2 raised to the power of 2. After second generation, the number of cells will be 4. Then after third generation, the number of cells will be 8. So n is equal to 3. So 2 into 2 into 2. So 8. So like that, the number of cells will increase by a factor of 2. So basically, it is increasing exponentially. Okay. Exponential increase means it is doubling after each generation. Okay. This is clear?
Yes, ma'am.
Okay. So we'll move to the next question. Question number 3. Which among the following is not a phase in bacterial growth? Stationary phase, lag phase, log phase, and dark phase. Which of the following is not a phase in bacterial growth? So you have a bacterial growth curve. Which of the following is not an actual phase that is used in the bacterial growth curve phases? So the
C.
sorry,
Ma'am, C.
log phase. You don't see log phase in bacterial growth. There are 4 phases in bacterial growth curve. The answer for this question is option D, dark phase. There is no dark phase in the bacterial growth curve. So if you see, here's the bacterial growth curve. So you have the lag phase, then the log phase or the exponential phase, then you have a stationary phase, and then you have the death phase. So the log phase is exponential phase is also known as log phase. So uh, in the lag phase, what is happening? The bacteria is adapting to its medium. Wherever you are allowing the bacteria to grow, it will first see its environment, it will adapt to the environment and start making different enzymes and start making copy of its DNA. So at that time, the bacterial number won't increase. After that, once the bacteria is ready to divide, it will start dividing, and I have explained how bacteria is dividing. So from one cell it is becoming two, then from two it is becoming four. So as the time will increase, there will be an exponential growth. Okay, exponential growth or a logarithmic growth. Okay, so the term log or the logarithmic or exponential, these can be used interchangeably. So log phase, so this is the log phase or the exponential phase. Okay, after exponential phase, the bacteria enters a stationary phase. So why it enters a stationary phase? So it will keep dividing and increasing in, like it will keep dividing and it will increase in number till the point where all the nutrients are available. Once one of the nutrients gets over, like the bacteria consumes all the nutrients, right? It is basically whatever medium it is growing, it is eating up all the nutrients and then it is growing. Once one of the nutrient is over, now it doesn't have enough food source to divide, so it will enter a stationary phase for some time, where it will use the energy from the stored food. So most of the microorganisms, they have storage granules and other type, and all the metabolites they have made during the exponential phase, so it will survive on those things, and it will keep, like it will not increase in number, but it will stay alive. Okay, so it will stay alive, it will utilize uh, the energy whatever is remaining. Once that is also over, it will start uh, dying. So the cells will start dying and there will be a decline in the number of live cells. So this is the uh, axis that is showing the number of live cells. So the number of cells will start decreasing after the stationary phase. So this is the death phase. Okay, so you have four phases: lag phase, log phase or an exponential phase, stationary phase, and then death phase. So there is no dark phase in this uh, bacterial growth curve. Okay, this is a very important curve, so you should understand what it is symbolizing. Is it clear?
Then what is in log phase? Log log. Like there's a
Yes,
lag.
this is the log phase. The exponential phase is also known as log phase. Okay.
Or when, like you can say the bacteria is not grow. In the dark phase is equal to the death phase. No.
No. Death phase is known as death phase only. It is not known as dark phase. There is nothing called dark phase in the bacterial growth curve. Okay. It's not the term that is used scientifically. Okay. The term exponential of the term logarithmic, these two are same. Okay, anything that is dividing continuously and doubling after each generation. So 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16. So like that, if it is increasing, that type of growth is called logarithmic growth or exponential growth, or logarithmic increase or exponential increase. Okay, so this is the log phase that a bacteria has. So lag phase, where it is adapting to the environment and thus number of bacteria does not increase. Log phase, the number of bacteria increases and it doubles after each generation time. Once all the nutrients are over, it enters the stationary phase, where again the bacteria number of bacteria is not increasing but it is still alive, it is not declining also. And then it enters the death phase, where all the bacteria starts dying slowly. Okay, clear?
Yes, ma'am. Why we use that phase is like this is used in Which conditions? I'm not understand this part.
Any bacteria when it is growing, so let's say you make a sugar solution, you add some salt, some nitrogen source, some phosphorus source, and make a solution, okay, make a medium, and then you put a bacterial cell, one cell of bacteria in that solution. So that solution is new for the bacteria, so it will first adapt to the environment, like what is there in the solution. It will start synthesizing all the enzymes that are required for growth. So at that time it will be in the lag phase. Once it is ready to divide, now it can just eat up all the nutrients and continuously divide till the point nutrients are available. Since it is eating up the nutrients, it will get over. Okay, it's a food. Okay, so if I am given some food, I'll keep on eating it till the food is there. Once the food is over, now it doesn't have anything to increase in number, so it will enter the stationary phase where it won't grow anymore, but it will still be alive by utilizing the energy from other forms of stored food, or let's say it made some metabolite on which it can survive. So it won't increase in number, it will become static. Okay, it will be alive but it won't increase in number. After certain point, now that it cannot survive anymore, it will start dying. So it is, this graph is basically showing the life cycle of a bacteria, how it starts growing and then how it starts dividing and then how it enters the death phase. So life cycle of one bacterial cell is defined by, one bacterial population is defined by this curve.
Okay.
Okay. So moving to the next question, question number 4. Any element that is least available relative to the requirements of an organism is defined as the dash, since the growth of the organism is dash. What will be the answer? So option A is limiting nutrient. So any element that is least available relative to the requirement of an organism. So it what will be the first term? Limiting nutrient, toxins, heavy metals, or maximizing elements. And the second is since the growth of the organism, since it is directly proportionate to the concentration of limiting nutrients, or since the growth of organisms are compromised in the presence of them, or since the growth of organism depend on the molecular weight of the metals, or since the growth of the organism is directly proportionate to the presence of elements. So which one of the following will be the correct answer? The correct answer for this question will be option A. So any element that is least available relative to the requirement of an organism is defined as the limiting nutrient, since the growth of the organism is directly proportionate to the concentration of the limiting nutrient. Okay. So what is limiting nutrient? So limiting nutrient is basically any so let's say you in a medium you have carbon source, nitrogen source, phosphorus source. So whichever source will be in the least amount. Okay, so let's say you have carbon source, 40% of carbon source, 30% of or 40% of carbon source, 40% of nitrogen source, and only 20% of phosphorus source, and you are making a bacteria grow in that medium. So what will happen, and that bacteria needs all the sources, like it needs carbon, nitrogen and phosphorus, all three of them. So what will happen? Phosphorus is less in amount, so naturally phosphorus is going to get over soon. So once the phosphorus is over, the bacteria won't be able to grow, because its growth is directly proportional to the concentration of the limiting nutrient. So the limiting nutrient is the one, like is that particular nutrient that is available in the least amount. So once that is over, the bacteria cannot grow, so its growth is directly proportional to the concentration of that particular nutrient that is in the least amount, that is present in the least amount. Understood? So this is limiting nutrient. So moving to the next question, question number 5. Identify the true and false statements from the following. First statement is selective media are used for the growth of only selected microorganisms. And second statement is differential media are used to distinguish one microorganism type from other another growing on the same media. So what do you think, the first statement is true or false? Selective media are used for the growth of only selected microorganisms. Is it true or is it false? So selective media are used for the growth of only selected microorganisms, and differential media are used to distinguish one microorganism type from the another growing on the same media. Are these statements true or false? So both of these statements are actually true. So selective media, they are used for the growth of selected microorganisms. So it is used to select between two microorganisms. So it will be a media in which only one type of microorganisms can grow. So you can select, like if you want any particular type of microorganism to grow on that, you will use that particular media that is selective for that microorganism. Okay, and differential media, on a differential media both the microorganisms can grow, but one microorganism will show some different properties than the other to distinguish between the two. Okay, so both of these statements are true statements. So selective media are used for the growth of only selected microorganisms. Media that inhibits the growth of unwanted microorganisms and support the growth of organism of interest are called selective media. Okay, so it will inhibit the growth of unwanted microorganism and support the growth of only organism of interest. Okay, and it can contain particular ingredients that will inhibit these growth of certain microbes. I'll give the examples now. And what is differential media? Differential media is media are used to distinguish one microorganism type from another growing on the same media. So both the microorganisms will grow in this case, but both will have different visually distinguishable appearance. So differential media contains compounds that allow group of microorganisms to be visually distinguished by the appearance of the colony or the surrounding media, usually on the basis of some biochemical differences between the two groups. Okay, so McConkey agar is an example of selective and differential media both. So it is selective for gram negative bacteria. So only gram negative bacteria can grow, gram positive bacteria cannot grow. So why is this the case? Why only gram negative bacteria can grow and gram positive bacteria cannot grow? So these McConkey agar, McConkey is the name of that particular agar. So in the McConkey agar, bile salts and crystal violet, it is a dye. So bile salts and crystal violet are present. So these two things, they actually inhibit the growth of gram positive bacteria. So in the previous session I have told you the structure of the gram positive and gram negative bacteria. So if you remember, gram positive bacteria had a thick peptidoglycan layer, but there was no outer membrane. In gram negative bacteria, the cell wall had a thin peptidoglycan layer, but it had an outer membrane outside the peptidoglycan layer. So that outer membrane actually inhibits the entry of bile salts and crystal violet, whereas uh, in gram positive bacteria, the bile salts can enter the gram positive bacteria since there is no outer membrane present, and it can actually kill the bacterial cells. That's why you can see Staphylococcus aureus, it is a gram positive bacteria, so there is no growth on the plate, whereas these are gram negative bacteria, and you can see the colonies of the bacteria. So bacteria are generally cultured on, when we have to grow bacteria on in the lab, we take these petri plates and then pour media on the petri plate. So we add agar to the petri plate, we add agar to the media, and then, so since agar is basically a solidifying agent, so when we pour the hot media on the petri plate, once it cools down, it gets solidified. So it's like a plate over which a jelly kind of a substance is present. Now that agar plate we can use to grow our bacteria of interest. So whatever bacteria we want to grow, we can just streak the bacterial colonies like this, and you can see bacterial colonies, small colonies will grow over the plate. So since McConkey agar inhibits the growth of gram positive bacteria, they will not grow, only gram negative bacteria can grow. So it is a selective media for gram negative bacteria. Okay, now how is it differential? The differential ingredient in this media is lactose. So other than bile salts and crystal violet, lactose is present, and a pH indicator called neutral red is present. Okay, so now it can differentiate between lactose fermenting and lactose non-lactose fermenting bacteria. So if a bacteria will ferment lactose, if it can use the lactose as the carbon source and ferment it into lactic acid. Okay, in the last class I have explained fermentation. So lactose will be converted into lactic acid. So once lactose is converted into lactic acid, what will happen? The pH will decrease. Since it's an acid, it will decrease the pH. That decrease in pH will cause the neutral red to turn into a bright pink or red in red color. So E. coli or Enterobacter aerogenes, these are all lactose fermenting bacteria. So you can see the color of the media becomes pink, the colonies and the color around the colonies, you can see it becomes pink. Whereas a non-lactose fermenting bacteria, when it will grow, it will create an alkaline pH, a basic pH, because of which the neutral color of neutral red will turn into yellow. Okay, so it's a pH indicator. So at a low pH the color will be pink, at a high pH the color will be yellow. So depending on the color of the colonies and color of the media, we can differentiate between lactose fermenting and non-lactose fermenting colonies. Okay, so this is how McConkey agar is both selective and differential. You understood the difference between selective and differential? So I have one more example. The second example is EMB agar or eosin methylene blue agar. Okay, so it is, the media is generally red in color. You can see the photo of this media. So it is also selective and a differential media. So it will also select, it is also selective for gram negative bacteria. So gram positive bacteria cannot grow on this media. Why? Because of this methylene blue, presence of methylene blue. So methylene blue, similar to bile salts and crystal violet, it will also inhibit the growth of gram positive bacteria, and only gram negative bacteria can grow over these media. And it can differentiate between lactose fermenters and non-fermenters. So in this media also lactose is present as the carbon source. If the bacteria is capable of fermenting lactose, this eosin dye will combine to methylene blue indicator to produce a color change when lactose or sucrose is fermented. Okay, so lactose fermenting coliforms such as E. coli produce blue black colonies with a green metallic sheen. So you see it looks very beautiful. You have the bacterial colonies and there is a green metallic sheen around the bacterial colony. So the colonies appear blue black in color and there is a green metallic sheen that is present around the colony. So this is how a lactose fermenting bacteria looks like. Whereas a non-lactose fermenting bacteria will only will look colorless. Basically the media is red, so it will appear red, but it will be colorless over the medium. Okay, so I hope you understood the difference between selective and differential media. So basically lactose fermenting bacteria will make lactic acid, and that will change the pH towards acidic, and at acidic pH this indicator will turn the colonies blue black in color and they give a green metallic sheen due to the amide bonding of the dyes in an acid solution. Okay,
Like ma'am, basically in that solutions, like in the differential media, in that differential media, like if you use lactose, what do you say, lactose agar, they can make the growth of bacteria. Like, but E. coli bacteria are like any positive bacteria, any gram positive bacteria. Sorry.
E. coli are gram negative.
Oh.
Okay, gram positive bacteria will not grow on this medium. Okay, so there are two terms, see, selective and differential. Differential means it can differentiate between two bacteria, so it can differentiate between lactose fermenting and lactose non fermenting. Okay, whereas
From your voice is breaking.
is it clear now?
Yes, ma'am.
So I was saying uh, there are two terms here, selective and differential. So selective means it can select one bacteria over the other. So it can select gram negative bacteria. Gram positive bacteria will not grow on this media. On both EMB agar also and McConkey agar, gram positive bacteria will not grow. So it is selecting, it is selectively allowing gram negative bacteria to grow. Okay, whereas differential means it can differentiate, it can distinguish between two colonies. So it can differentiate between lactose fermenting and non lactose fermenting. So bacteria that can So both of these bacteria will grow on this medium. Gram positive will not grow on this medium. So basically it is selecting gram negative and then differentiating between gram negative that can ferment lactose and gram negatives that can not ferment lactose. Okay.
Ma'am, this is a basically solution like
It's
this.
a solidified media. So it's a solution in which we add agar. So agar is a solidifying agent. So what happens? Agar is a powdered compound. So when we add agar and when we heat the solution, the agar basically polymerizes, and then once the solution is cooled down, it solidifies. So it's a Petri plate. Do you know what a Petri plate is?
Yes, ma'am.
Huh. So on a Petri plate, we pour this hot media, okay, and we let the Petri plate to cool down. So once it cools down, it solidifies in that Petri plate, okay. So it's like a bed of jelly, okay,
Okay, ma'am.
like it's gel kind of a thing. So it's a gel pad on which we streak the bacterial colonies. So we have pure cultures of bacteria, and then we can take those bacteria and just streak it over the solidified medium and then incubate the plates, like keep the plates at 37 degrees overnight to let the bacteria grow. Once the bacteria grows, we will see, like after the overnight time period, we will see if the bacteria has grown or not. If it has not grown, that means it was gram positive. If it has grown, that means it is gram negative. Now once it has grown, we will see what color is the what color does the colonies have. If the colonies have pink color, that means it is a lactose fermenting gram negative bacteria. If the color of the bacteria, like if the color of the colonies is colorless or the media is yellow in color, that means it is non lactose fermenting gram negative bacteria. Okay. Is it clear now? Okay. So we'll move to the next question, question number 6. Match the following uh, temperature ranges with the microbial growth. So we have different temperature ranges starting from -30 till 130 degrees Celsius, and we have different different types of microbial growth mentioned. So we have to match the two. So let's see, -30 to 0 degrees Celsius. Which of the following microbial growth will match with this -30 to 0 degrees Celsius? Do you think there will be any microbial growth in this temperature range? So there will be no significant microbial growth below this point, because at this temperature it is very difficult for a bacteria to survive, since at a lower temperature all the enzymes become non-functional and the membrane also starts gelling up, because the membrane is a I have told you the membrane is plasma membrane is basically phospholipid bilayer, right? So it's a it has a fluid mosaic structure, right? So that fluidity at a lower temperature, it will become less fluid, it will become gel like, so because of which the bacteria cannot survive. So there will be no significant growth at -30 to 0 degrees Celsius. So A will be F. Then 0 to 5 degrees Celsius, at 0 to 5 degrees Celsius it is basically the temperature of the refrigerator we have. Okay, so refrigerators generally have 0 to 5 degrees Celsius, and it may allow slow growth of spoilage bacteria and very few pathogens. So you must have seen we store whatever leftover food or whatever products are there in the refrigerator at home, right? Why do we store food in the refrigerator? To slow down the growth of spoilage bacteria. Okay, so if you leave any cooked food outside, it will get spoiled very quickly because all those bacteria have optimum temperature to grow. So when you are putting the food inside the refrigerator, what you are doing, you are slowing the growth of these spoilage bacteria. So but if you will leave the food for a very long time inside the fridge, it will still get spoiled because these bacteria are not dying, they are only growing slowly now. So it will just elongate the shelf life of the food. Okay, then next is, so for B it will be A, so A F B A. Then C is 5 to 16 degrees Celsius. So 5 to 16 degrees Celsius, this is the temperature range where many bacteria survive and some may grow. Okay, so in the last class also I have told you there are certain bacteria that can survive at lower temperature or very higher temperatures. So 5 to 16 degrees Celsius, some bacteria can grow, and most of the bacteria, they can survive, they can tolerate that temperature. Then 16 degrees to 52 degrees, this is the temperature range where rapid growth of bacteria is observed, and some bacteria can produce toxins at this temperature. So 16 to 52 degrees Celsius, it is assumed to be the optimum temperature for a bacteria to grow. Then 52 to 62 degrees Celsius, very slow bacterial growth, because again the bacteria is entering the extreme conditions. And from 62 to 130 degrees Celsius, the temperature in this range destroy most microbes, although lower temperatures take more time. So I have told you process of sterilization is done at high temperatures. So process of sterilization, that autoclaving, is generally done at temperatures near 130 degrees Celsius. Okay, so autoclaving is done at 121 degrees Celsius. So it is almost up to 130 degrees Celsius. All bacteria and microorganisms are killed. Okay, from 62 degrees Celsius onwards, the bacteria starts getting destroyed. Okay, so at a lower temperature, it will take more time to kill all the bacterial cells. At higher as you will increase the temperature, obviously the time taken to kill the bacterial cells will decrease. Okay, so the correct answer for this question is option number C. So here if you see how does the temperature affect the microbial growth, you can see below 0 degrees Celsius, most of the enzymes work slower at the colder temperature, and I've told you about membrane gelling. So there will be very small there'll almost be no growth in this region. Then from 0 to 5 degrees Celsius, slowly the bacteria will start growing, and then from 5 to 16 degrees Celsius, again there will be most of the bacteria will survive, but growth will not be significant growth will not be there. Then from 16 degrees to 52 degrees Celsius, most of the bacteria can rapidly grow. So you can see there is an exponential increase here. And then from 52 to 62, again the bacteria starts decreasing in number. So you can see at 16 to 52 degrees Celsius, the enzymes work best at this temperature. So around 37, our body temperature is 37 degrees Celsius. So around that temperature only most of the bacteria uh, grows rapidly. From 52 to 62, the bacteria starts dying, and after 62 degrees Celsius, most of the enzymes denature at this higher temperature, and since enzymes are non-functional, most of the bacteria also cannot grow and survive at this higher temperature. Okay, this is clear. So we'll move to the next question, question number 7. Bacteria that have an optimum temperature of 35 to 39 degrees Celsius are called: option A, psychrophiles; option B, mesophiles; option C, thermophiles; and option D, psychrotrophs. So what is the bacteria called if it has an optimum temperature of 35 to 39 degrees Celsius? Anyone knows the answer? Yes, option B, mesophiles. So the correct answer is mesophiles. So mesophile is an organism that grows best in the moderate temperature, neither too hot nor too cold. So 35 to 39 degrees Celsius, it's a moderate temperature. Okay, it's neither too hot and neither too cold. Psychrophiles are extremophilic bacteria which can live at colder temperatures. So these psychrophiles, the term philes means loving. So psychro means at a lower temperature. So cold loving bacteria, they have the optimal temperature for growth at about 15 degrees Celsius or lower. So it generally grows at the lower temperature, and the minimal temperature for growth at 0 degrees Celsius or lower. Okay. Then thermophile are again extremophiles that can thrive at high temperatures. So from 41 degrees Celsius to 120 degrees Celsius it can survive. So these are these are the bacteria that can survive in extreme cases or extreme conditions. Okay. Psychotrophic bacteria, these are different from psychrophiles. Psychrophiles can grow at 15 degrees Celsius or lower, but psychotrophic bacteria that can grow at 7 degrees Celsius, although their optimum growth temperature is higher. So it can tolerate the lower temperatures, it can grow at lower temperatures also, but generally if it has an option it will prefer the higher temperature, higher growth temperatures only. Higher means around mesophilic temperature. Okay. So these 4 terms are clear, I hope. Mesophile, psychrophile, thermophile and psychotrophic. Okay. So moving to the next question. What is the upper temperature limit for growth of algae and insects? Option A is 38 degrees Celsius for algae and 40 to 45 degrees Celsius for insects. Option B is 50 to 60 degrees Celsius for algae and 60 to 62 degrees Celsius for insects. Option C is 45 to 50 degrees Celsius for algae and 55 to 60 degrees Celsius for insects. Option D is 55 to 60 degrees Celsius for algae and 45 to 50 degrees Celsius for insects. So what is the upper temperature limit for growth of algae and insects, we have to tell. So the correct answer for this question is option D, 55 to 60 degrees Celsius for algae and 45 to 50 degrees Celsius for insect. Okay. So this is a theoretical question, you can just remember the temperatures. Moving to the next question, question number 9. The ratio between the vapor pressure of a food item when in a completely undisturbed balance with the surrounding air media and the vapor pressure of the distilled water under identical condition is called. What is the ratio between the vapor pressure of a food item at a particular environment versus the vapor pressure of the distilled water under the same environment is called? Option A, water activity. Option B, evaporation rate. Option C, moisture content. And option D, saturation point. So what will be the answer? The ratio between vapor pressure and vapor pressure of the food item and the vapor pressure of the distilled water under the same conditions. What will be the answer? Anybody? The correct answer for this question is option A, water activity. So what is water activity? Let's see. So water activity of a food, water activity is generally defined, written in terms of AW. Water activity of a food is the ratio between the partial vapor pressure of the food itself when in a completely undisturbed balance with the surrounding air media and the vapor pressure of the distilled water under identical condition. So that means a water activity of 0.8 means the vapor pressure is 80% of the pure water. Okay. Water activity increases with the temperature and the moisture condition of a product can be measured as the equilibrium relative humidity expressed in the percentage or as the water activity expressed as the number ranging from 0 to 1. Just a second. So, why do we use this term water activity? Where do we use this term water activity? To see how well a microorganism can grow. So, water activity is actually related to the ionic strength of the solution. So, in pure water, the ionic strength is very low, almost negligible, because the conductivity of pure water is almost equal to 0. Okay, so there are no salts present in the pure water. By pure water I mean double distilled water. Okay, so in pure water, since the ionic strength is very low, so it is used kind of like a scale, like a standard. So if the ionic strength as the ionic strength keeps increasing, the water activity keeps decreasing for the particular medium. So water activity of pure water would be 1. Okay, since the ionic strength is almost 0. Then the water activity of other uh, food items or any medium, it will be lower than the water activity of the uh, pure water, as it will have some forms of ion that can be used by the microorganisms to grow. Okay, so lower the water activity, higher will be its ionic strength. Okay, this is clear, I hope. So moving to the next question, question number 10. True or false? We have to state whether the two statements are true or false. First statement is bacteriostatic inhibits growth, they do not kill cells. And the second statement is bacteriolytic kills bacteria but do not cause cell lysis. So is the first statement true? Bacteriostatic inhibits growth but they do not kill the cells. Is this statement true or false? So it is a true statement. Bacteriostatic, it inhibits growth, but it does not kill the cells. So it's a true statement. So one is true. And then bacteriolytic kills bacteria, but do not cause cell lysis. Yes, the option A is the correct answer. You're right. So this statement is false, since in the name only you can say bacteriolytic. Lytic means it can cause the cell lysis. It can lyse the cell. It can break the cell. Okay. So first statement is true. Second statement is false. So bacteriostatic compounds, they can inhibit the growth, but they do not kill the cells. Example is refrigeration. So I've told you we keep the food item inside the refrigerator to slow down, to inhibit the growth of the bacteria. We do not kill the bacteria by refrigeration. Again, once you will take the food out of the refrigerator, the growth of the bacteria will resume to normal levels. Okay. So this is the graph that is explaining the growth of bacteria when a bacteriostatic agent is applied. So once a bacteriostatic agent is applied, is the growth is not decreasing, the growth is stationary. It is just inhibited, so it is not increasing, neither it is decreasing. Okay. Bactericidal compounds kills the bacteria, but they do not cause cell lysis. Example is antibiotics. So they kill the bacteria, but they do not cause cell lysis. They do not break the cells. So since they are killing the bacteria, the growth will decrease, but total number of cells, like if you, by total number of cells we mean number of live cells plus number of dead cells. So total number of cells will be same, because the cells are not breaking down, the cells are not breaking into cell debris. So number of cells will be constant, only the number of live cells will decrease. Whereas in bacteriolytic uh, agent, it kills the bacteria by causing cell lysis. So both total number and the viable cell number will decrease in number. So it is done by using chlorine or ozone. Okay. So the next question is question number 11. Which of the following term refers to an agent that inhibits bacterial growth? Which of the following term refers to an agent that inhibits bacterial growth? Option A is fungicidal, option B is homeostatic, option C is bactericidal, option D is bacteriostatic, and option E is germicidal. So the correct answer is yes, option number D. So option number D, bacteriostatic, it refers to an agent that inhibits the bacterial growth. I have already explained bactericidal, bacteriostatic, and bacteriolytic. We will see what the other terms fungicidal, homeostatic, and germicidal means. The correct answer is D, bacteriostatic. Fungicidals are the compounds that selectively eliminates the fungal pathogens. So in the name, fungicidal is there, so it will selectively kill the fungal pathogens from a host with minimal toxicity to the host. The term homeostasis, it means self-regulating, like it's a self-regulating process in which an organism tends to maintain stability while adjusting to conditions that are best for its survival. So our body is our body maintains a homeostatic condition where whatever the temperature outside is present, like even if it is winters or summers, our body is always at a constant 37 degrees Celsius. Okay. And germicidals are substances that can kill any form of germs. So Here by germs we means any form of microorganisms that can cause infection or disease. Okay, so it's a generalized term, whereas bactericidals are the particular terms that particular substances that are used to kill only bacteria causing infection. Okay, so moving to the next question, question number 12. Which of the following is not a characteristic of the autoclave? So we have to tell which of the following is not a characteristic of autoclave. Option A is the ability to sterilize solutions with endospores, the requirement for long exposure time, the use of moist heat, and option D is the use of high pressures. So which of the following is not a characteristic of autoclave? So ability to sterilize solution. Can autoclave do that? Autoclaves cannot sterilize uh, sorry, autoclaves does have the ability to sterilize endospores. Since the autoclaves reach the temperature of 121 degrees Celsius, so it can kill the endospores that are that form that are present in the medium. Use of moist heat, autoclaves use steam sterilization technique, so it is using moist heat and it is done at high pressures. So the characteristic that is not applicable to autoclave is requirement of long exposure time. So what is an autoclave? I'll explain first. So it's a device which is used to sterilize any medium or any component uh, you want to get rid of all microorganisms and endospores. So you have a lid which has a pressure gauge and an adjustative pressure valve, and then inside the it's a stainless steel container, and inside this container you have a perforated metallic baskets, um, in which you can keep your stuff whatever you want to sterilize. And then there is so this is a double jacketed layer, one metallic basket and then the outer stainless steel vessel. So between these two we fill water. So the water is almost at the base of this metallic basket, and then this water has heating coils applied to uh, like heating coils attached to the instrument, and these heating coils can heat up the water and steam will get generated here in this chamber where your uh, things are kept for sterilization. And then that steam under pressure it will reach up to the temperature of 121 degrees Celsius. So at 15 psi pressure, since the volume is constant, at 15 psi pressure the autoclave uh, temperature reaches 121 degrees Celsius, at which all the microorganisms and the endospores are killed. Okay, and then once the so this process is done for 15 to 20 minutes, so it's the exposure time is not very long. Okay, so this is how an actual autoclave looks like. So the principle of an autoclave is very similar to the pressure cooker that you have at your home. So under under high pressure what happens is the temperature the increase in temperature happens quickly. Okay, so if I keep boiling the water it will only reach till 100 degrees Celsius because the boiling point of water is 100, right? But if I apply pressure at a constant volume, what will happen? The temperature will reach up to, like the temperature will increase more than 100 degrees Celsius and it can reach up to 121 degrees Celsius at 15 psi pressure. So you can see the pressure from the pressure gauge, and the point where the pressure has reached 15 psi, you are supposed to maintain the pressure at that point for 15 to 20 minutes, so that the things that you have kept inside the autoclave, it will it will remain at 121 degrees Celsius, that is achieved at 15 psi pressure. For 15 to 20 minutes it will be exposed to the 121 degrees Celsius temperature, 121 degrees Celsius steam, and all the things will be sterilized, as all the microbes will be killed. Okay, so this is clear, I hope, how an autoclave works. So moving to the next question, question number 13. Again, true or false? Uh, Two statements are there. We have to tell both the statements are true or false. UV and X-ray radiations are similar in that they damage DNA and have high penetrating power. And second statement is most disinfectants can be used as antiseptics. So first statement is true or false? UV and X-ray rays are similar in that they damage DNA and have high penetrating powers. Is it true or is it false? So Amardeep has said option number B. So the first statement is false. And most disinfectants can be used as antiseptics. It is also false. So yes, the correct answer is B. Both the statements are false. So UV and X-rays both can cause DNA damage, but UV has a lower frequency and longer wavelength, whereas X-ray has a higher frequency. So UV with its longer wavelength and less energy is less penetrating than X-rays. Okay, so both of them are not having high penetrating power. So X-rays have the potential for destruction of DNA and sterilization. Both UV and X-rays can damage DNA, but penetrating power of X-rays is much higher than UV radiations. Okay, and about the second statement was most disinfectants can be used as antiseptics. So disinfectants are used to destroy or remove vegetative pathogens, while antiseptics are used for destruction or inhibition of microorganisms on living tissues. So I've told you in the first question only what is the difference between the disinfectants and the antiseptics. So disinfectants can destroy the vegetative pathogens. So any surface, if it is wiped with a disinfectant, it will just remove the vegetative pathogens that is present there, whereas antiseptics, it is used to destroy or inhibit the microorganisms on the living tissues. So I've told you, if we get any wound on the skin, we generally wipe it using Dettol solution. So Dettol is nothing but an antiseptic liquid which can kill the microorganisms on that living tissue, and it can uh, inhibit the growth of any new microorganism. Okay, even antibiotics are given in this case. Okay, so this is clear. So we'll move to the next question, question number 14. Which of the following method is bactericidal? Deep freezing. Option B is lyophilization or freeze drying. Option C is membrane filtration. And
C.
option D is, sorry, you said option C.
Yes.
Option C is bactericidal. Membrane filtration, can membrane filtration kill the bacteria? Bactericidal means an agent that can kill the bacteria. So the correct answer for this question, yes, the correct answer for this question is option number D, addition of strong acids. So strong acids can kill the bacteria. Addition of strong acid, or in that case addition of strong base, can kill the bacterial cells. Whereas all these three methods, it won't kill the bacterial cells. So strong acids can destroy cells without causing cell lysis and are therefore bactericidal. Okay. The other three methods mentioned do not kill the cells. Deep freezing and freeze drying are bacteriostatic. But what is bacteriostatic? I have told you, it can just inhibit the growth of cells. Okay, the cells won't die, the cells will just enter a dormant phase where they cannot grow and increase in number. Once they are taken out of the freezer, again they will resume their normal growth after some time. Okay, so both deep freezing and freeze drying, these are bacteriostatic methods. Membrane filtration is considered a disinfection method because it can remove microbes like protozoa, fungi, bacteria and viruses. So membrane filtration can be considered a disinfection or sometimes a sterilization method also. So what membrane filtration is doing, it is just, it's like a filter. Okay, so it is just uh, separating the microbial cells from the rest of the media components. Okay, so it's a paper with, or a cellulosic membrane, or like any kind of polymeric material can be there for a membrane. So it can be made of some nylon membrane, nylon material, or some kind of polymer. Okay, so that polymer will have certain uh, that polymer will have certain number of pores present of a particular pore size. So membrane filter having that particular pore size, so whatever components are bigger than the pore size, they will remain over the membrane and rest of the things will get out of the membrane. So it's like how you filter your tea from the tea leaves, right? You take a sieve and you pour the tea with the tea leaves, and the tea leaves, since they are bigger than the pore size, they remain over the sieve and the tea gets filtered out, and that's how you separate tea from the tea leaves. So similarly, you separate uh, different microbial cells from the rest of the media by using a filter. So filter is not killing the cells, it is just separating, it is a physical separation method. Okay, I hope this is clear. Is this clear, Remika? Why membrane filtration is not bactericidal?
Yes, ma'am.
Okay. So moving to the next question, question number 15. Antimicrobial activities can be measured using option A, spread plate method, option B, pour plate method, option C, disc diffusion method, and option D, membrane filtration method. So the correct answer, yes, option C. Option C, disc diffusion method. So antimicrobial activity can be measured using disc diffusion method. What is disc diffusion method? In this method, paper discs are soaked in a solution of disinfectants or the antimicrobial agents. So like this, small paper discs are taken and they are soaked in different antimicrobial agents and placed on the surface of the nutrient medium, on which a culture of a test bacteria has been spread to produce uniform growth. The antimicrobial activity of the different chemicals can be measured by measuring the zone of inhibition around each disc. So what is done? You take a petri plate with a nutrient medium, agar, and you spread the culture all over the agar, so that it will grow uniformly. And then just after spreading, we take this small disc soaked in different so here you can see it is soaked in different antibiotics. So antibiotics are also antimicrobial agents. So this is soaked in vancomycin, this is soaked in ampicillin, this is soaked in I think methicillin. So different antibiotics, these discs are soaked and then placed over the plate, and then the plate is allowed to incubate, allowed to the plate is kept at 37 degrees Celsius and the bacteria is allowed to grow. So you can see all this uh, lawn formation. This is due to the growth of the bacterial cells. And you can see there is a zone of inhibition around each disc. So since the discs have the ampicillin or vancomycin soaked in it, what will happen that from the disc, this ampicillin or vancomycin or whatever the antimicrobial agent is, it will spread, it will diffuse in the agar radially. Okay, so it will diffuse in the agar in every direction. Since it's a circular disc, the diffusion will occur radially. So and the whatever the antimicrobial power of each component will be. So here you can see methicillin has more uh, uh, methicillin has more antimicrobial power than ampicillin. So you can see the zone of inhibition is bigger for methicillin than ampicillin. So antimicrobial activity of methicillin is higher for this particular bacteria. Okay, so like that we measure the zone of inhibition, we measure the diameter of this zone of inhibition, this circle that is forming. So what is happening, since the ampicillin is diffused in this area, the bacteria is inhibited by the ampicillin and it cannot grow, and thus this clearance is there. Okay, so in this area there is no bacterial growth, in this area there is no bacterial growth. Okay, and here where no antibiotic is present, bacteria has grown happily, and you can see the long lawn formation here. This is clear how zone of inhibition is measured and what is the disc diffusion method. So this method can be used for any antimicrobial agent. Yes, any question?
Ma'am, this is a van AMP control. Like this is something. This is like the paper, or it's
Yeah,
look like the paper.
this is from one of the papers. So
Okay,
this is
okay, okay.
I have just added this. I'll add the reference
Mm-hmm.
here. So I've
Okay,
just
okay.
added this image for explaining what disc diffusion method is. So
Ma'am, like this, the circle is form is outside. Means how many distance, how many diameter of that. That is also be like we have to be think when we are go for the antimicrobial, like that much
yes, so
diameter
that is what the diameter of this
is bigger.
inhibition,
I think
it is,
nith something.
so it is defining the antimicrobial activity of this compound. So all these small paper disc, this white colored thing, this is basically paper. Okay, small filter paper, circular,
Yes.
which is like a small piece of filter paper is taken, cut into a circular piece, and then it is soaked in the antimicrobial agent. So this is one antimicrobial agent, this is second antimicrobial agent, and this is the third antimicrobial agent, and this is the control which is just soaked in, let's say, water. So then what happens, we just place the disc over the bacterial colonies and see up to what point the bacteria can grow. So since the antimicrobial activity of this particular compound is higher, you can see the bacteria is inhibited more in this region. The diameter of this zone of inhibition is bigger, right, than these two. So that means the bacteria cannot grow in this area. Whereas for this particular compound, the bacteria could grow more closer to the disc than this particular compound. Is it clear now?
Yes, ma'am. Ma'am, it's the diameter of that distance between that circle
Yes.
and that the paper, if is it more, means the inhabitations would be growing good.
Inhibition
Like,
is more. Yes, inhibition
okay,
is more. Yes.
okay. Thank you, ma'am.
Okay, so this is the disc diffusion method. So once you will see the growth of bacteria, so what uh, higher the antimicrobial activity, the larger will be the zone of inhibition around the disc. Okay, it is called disc diffusion because the compound is diffusing in the agar plate, and then inhibiting the bacteria to grow around the disc. Okay.
The ma'am one use one word MIC, minimum inhabited something.
Minimum
MI,
inhibition concentration, MIC.
yes.
Yes.
That means of same, ma'am. The mean of whatever you can tell, it's means of same like this. How many zones would be?
Minimum inhibition concentration would be the minimum concent. So from this you can just see which is which compound is inhibiting more and which compound is inhibiting less. Okay, but to uh, calculate the exact concentration of the compound, you have to do another experiment where you will take different concentration. So that also you can do with disc diffusion. So you can take small paper discs and you can soak in the same compound. Let's say I have to check the MIC of ampicillin, this particular compound. Okay, so I'll make different ampicillin solutions ranging from concentration of 0 to concentration of 100 mg per ml, let's say. Okay, and soak the I'll soak the paper disc in different ampicillin solution of different concentrations, and then similarly I'll spread the plate like this, and then I'll keep the different ampicillin disc of different concentration all over the plate, and then see at what particular concentration the inhibition is starting. So you will see at 0, that is the control, there will be no inhibition since there is no ampicillin. Then at a concentration of, let's say, very 10 microgram or 10 nanogram per ml, there might not be any inhibition in that case also. Then at a concentration of 20 nanogram per ml, again there will be no inhibition. So like that we will see at what particular concentration the inhibition is starting. So that will be the minimum inhibition concentration, the minimum concentration required to inhibit the growth of bacteria. So the concentration at which the zone of inhibition will start, this is called the zone of inhibition. This clearance, it is called the zone of inhibition. So the concentration at which that particular zone will start, that is the concentration that can be defined as the minimum inhibitory concentration. Okay, clear?
Yes.
Okay, so moving to the next question. Question number 16. Decimal reduction time is the time in minutes in which dash of the population at a given temperature will be killed. Option A 75%, option B 10%, option C 100%, and option D 90%. So what is the percentage of population that should be killed in one decimal reduction time?
90.
Yes, the answer is option D, 90%. So decimal reduction time, it is defined as the time required to kill 90% of the microbial population exposed to specific temperature, or the time at which only 10% of the original population is remaining after exposing to specific temperature, It is the decimal reduction time, or it is also known as the D value. Okay, so either you can see how much population is killed, or you can see how much population is remaining. So either 90% population should be killed, or you can say 10% 10% of the original population is remaining. Okay, so if you start with 100 cells and expose it to specific temperature, so the time taken to kill 90 cells out of those 100 cells will be the decimal reduction time. Okay, or the time taken up to only 10 cells are remaining from out of those 100 cells is the decimal reduction time, or the D value for that particular cell at that particular temperature. Okay. So question number 17. All of the following methods are used for food preservation except option A, commercial canning, option B, adding sugar, option C, vacuum packaging, and option D, pasteurization. So out of all the following options, which of the option is not used for food preservation? Basically, yes, Amardeep has answered option B, adding sugar. So basically we have to say which of the following options are uh, out of the following option which cannot prevent the microbial growth. So the correct answer is adding sugar. So addition of sugar actually helps in preserving the color, texture and flavor of the food, but it does not inhibit microbial activity, so it cannot be used for food preservation. So most of the commercial food you have seen, it is uh, sold in form of canned. So you have canned peas and canned beans. So this is commercial canning. In those tin cans, it is sorry, in those tin cans the food is stored and sealed in such a way that it can inhibit the growth of any microbes inside. Then vacuum packaging, since you know that most of the microbes require air or some or the other nutrients, so in the vacuum there won't be any nutrient present. So the in this way also you can inhibit the growth of microorganism. Pasteurization I've already explained previously. So it is a type of preservation method for milk, mainly used for milks. So addition of sugar, it is also used in the food products. So most of the jam and other uh, juices that you get, the packed juices, they have high amount of sugar, but that sugar is added to preserve the color, texture and flavor of the food, but it cannot inhibit the microbial activity, because microbes can use the sugar as the carbon source and it can grow. Okay, so this is clear. So moving to the next question, question number 18. Which of the following is suitable for use of tissues for microbial control to prevent infection? Which of the following is suitable for use on tissues for microbial?
Antiseptic. B.
Yes, antiseptic. Yes, both of you are correct. So option A is disinfectant, option B is antiseptic, option C is sterilant, and option D is water. So answer is option B, antiseptic. Use of chemical antimicrobials for destruction or inhibition of microorganisms on living tissue is called antisepsis, and the chemical used is called antiseptic. I've already explained about this earlier. So the next question, question number 19. Filters having pore size of dash are often used for sterilization. Option A 0.08 micrometer, option B is 0.45 micrometer, option C is 0.22 micrometer, and option D is 0.03 micrometer. So what is the pore size that is often used for sterilization? Now for sterilization we want to remove all the microbial cells. So what is the pore size that should be used?
Ma'am, C 0.2
Yes,
microns.
0.22 microns. So why do we use 0.22 microns?
Ma'am, because the size of that bacteria is very very small. That's why we can use it that size of
Yes.
filter. I
So out of all the microorganisms, size of bacteria is the smallest, uh, like apart from viruses. We are not considering viruses here because it cannot uh, grow independently anyway. So out of other microorganisms, bacteria have the smallest size range, and the size range for bacteria is 0.3 to 0.5 micromolar. So if you use any pore size less than 0.3, you can actually remove the bacteria from the bacterial cells, and obviously if you can remove the bacterial cells, you can remove the other microorganisms also from a medium. Okay, for removing viral cells you have to use further smaller zero point 0.1 micron range. You have to use the filters. Okay, filters of pore size of 1, 0.1 micron range or lower than that. Okay, next question is, which of the following category of oxygen tolerance is depicted in tube 3? So here are 5 tubes showing different oxygen tolerance. So in the tube 3, which category of oxygen tolerance is shown? Option A is aerobic, option B is facultative aerobes, option C is anaerobic, and option D is microaerophilic.
Ma'am, I think B. I'm not sure. It was little confusing that question.
Option B, facultative aerobes. So yes, the correct answer is for facultative aerobes. So let's see what are all these 5 categories. So in the first category, so it's basically a tube containing any medium in which the bacterial cells are growing. So if the bacterial cells are growing near the surface, that means it requires oxygen. So in this first tube you can see all the cells are present near the surface. None of the cells are present at the bottom surface. So you know oxygen can penetrate only to short distances. So only obligate aerobes that require oxygen, that cannot grow without presence of oxygen, they can only grow in such conditions. So obligate aerobes will be present only in this region. Okay, then in the second tube you can see all the cells are present at the bottom of the tube, which is the most anoxic zone, because oxygen cannot penetrate till this bottom of the tube. That means these uh, cells, they are sensitive to oxygen, and that's why they are moving away from oxygen and growing at the bottom of the tube, and these cells are called anaerobes or anaerobic cells, right? Then the third is facultative aerobes. So facultative aerobes, they can grow both in presence or absence of oxygen. So that's why you can see the growth is throughout the tube. All the cells are present throughout the tube. But if you see, the number of cells at the surface is more than the number of cells present uh, in the anoxic zone. Why? Because the growth is better near the surface, because these organisms can respire. So they can grow better in presence of oxygen, but they can also grow in absence of oxygen as well. Okay. Then fourth is microaerophilic. So microaerophilic or microaerophiles, they can grow away from most of the oxic zones. So they can basically tolerate very small amount of oxygen. So they are growing little bit away from the surface. So let's say the oxygen will be reaching this point, so it is growing little below that region. So micro aerophilic, by the name you can see it can tolerate very small amount of air or the oxygen. Okay, and then the last is aerotolerant anaerobes. So these are mostly anaerobic bacteria but they can also tolerate air, they can also tolerate the oxygen levels. So that's why the growth is throughout the tube. But you can see the difference between third and fifth tube, the growth is very dense at the surface of the tube, although the growth is throughout the tube in both the cases. In this case, at the surface the growth is not that dense because these uh, aerotolerant anaerobes, these are mostly anaerobic in nature, so they grow in the anoxic zone only. But they can, since they can tolerate the oxygen levels, how they are tolerating the oxygen level, they can use the oxygen by doing fermentation process. So in the last class I have explained how uh, microorganisms can do fermentation. So they can grow in presence of oxygen, like they can tolerate the oxygen but then they cannot grow in presence of oxygen, they can only do fermentation. So that's why the growth is throughout the tube, but mostly they prefer the anoxic zone. Okay, so first is aerobic or obligate aerobes, second is anaerobic, third is facultative aerobes, fourth is microaerophilic, and fifth is aerotolerant anaerobes. Clear? All five terms. This is a very important, like these five terms are very important to define any bacteria, nature of bacteria. So this is clear?
Clear.
Okay. So the next question is, which phase has the condition of specific growth rate mu is equal to 0? So I have told you about the bacterial growth curve. We had four phases, right? Lag phase, log phase, stationary phase, and death phase.
A. Then answer is A because it's a lab phase. The all thing would be started. I think.
Yeah. So the question is, at which phase the specific growth rate will be 0? So at lag phase, the cells are preparing to grow. So the growth rate will not be 0. It will be very close to 0, but it won't be equal to 0. At log phase, it is obviously not 0. It is increasing in number. At stationary phase also, the specific growth rate will not be 0. The specific growth rate at stationary phase will be at the maximum level. So at death phase, the specific growth rate will decrease to 0 because now the cell number is decreasing. Okay, so the answer will be death phase, since in the death phase the cell lose viability and they start lysing. Lyse means they start breaking, bursting. Okay, so therefore mu is equal to 0 at death phase. So if you remember, I have shown you the growth curve, right? So in the growth curve, we had the lag phase, log phase, then the stationary phase, and then death phase. So in all three of them, lag, log, and stationary, the cells are uh, either increasing in number or increasing in weight. But in death phase, the cell number and cell weight both is decreasing because the cells are lysing now. Cells are bursting and breaking down. Okay, so cell number will decrease and thus the specific growth rate will be 0. Okay. Question number 22. Lag phase constitute of dash. Option A, no change in number but an increase in mass. Option B, change in number but decrease in mass. Option C, no change in number and decrease in mass. And option D, constant number and mass. So lag phase constitute of what?
B.
B, change in number but decrease in mass. In lag phase, did you see any change in number? So the correct answer is no change in number and increase in mass. So this is the growth curve. So I'll explain both the questions, the previous question and this question also. So in the lag phase, I've told you the cells are now preparing to grow. So cells will start synthesizing all the enzymes and essential nutrients that are required for the log phase. So it will prepare itself for the log phase. So there will be no increase in number. Number of bacteria is constant, but it will increase in mass. How it is increasing in mass? Because it is synthesizing all the enzymes inside the cell. So the cell mass will increase, but cell number will remain same. Okay, so this is the lag phase. After the lag phase, the cells will enter the log phase or the exponential phase, where the cells will continuously divide and increase in number exponentially. That means it will double after every generation time. So one cell will become 2, 2 cells will become 4, 4 cells will become 8, and so on. It will increase in number till the point nutrients are present in the environment or in the medium. Once nutrients are getting over, it will enter the stationary phase. So at stationary phase, it has reached the maximum growth rate. Okay, so after reaching the maximum growth rate, there is no further growth possible for this bacteria. It will enter the stationary phase, where it will survive on some other metabolites and stored foods that that it has produced. Once everything is over, the cells will start dying by lysis, and the uh, cell number will start decreasing. So this is the part where the specific growth rate is 0, because now the cells are losing the viability and the cells are getting lysed. Okay, so this is the lag phase. Lag phase is the time period where there is no immediate increase in cell number of the microorganism as they are introduced into the fresh culture medium. Microorganisms during this phase are trying to acclimatize to the new culture medium. They are trying to adjust to the new culture medium. They are trying to adapt. Okay, as it may be different from the previous one in which the microorganism was growing. So if I am taking a microorganism from soil and adding to a synthetic medium, so it will take some time, because the environment in the soil was different than the medium I have added it. So it will need to prepare itself. So that preparation happens in the lag phase. Typically, cells in the lag phase are synthesizing RNA, enzymes, and essential metabolites that might be missing from their new environment, such as growth factors and macromolecules, as well as adjusting to the environmental changes such as pH, temperature, oxygen availability, etc. They can also be undertaking any necessary repair of injured cells. So once I am taking a bacteria from soil and adding it to the medium, it will start synthesizing all the different enzymes and metabolites that are required, that are missing in the new environment. So it had some different enzymes in the soil, but now in the new environment it requires some different enzymes. So it will start synthesizing them, and it will start adjusting to the new temperature, the new pH. The soil pH must be something different, and the pH I have inoculated it in is something different. So though, and then if some cells are injured, so they will repair those cells. So all those things happen in the lag phase. So in the lag phase, the cell number do not increase, but the cell mass can increase since all the synthesis is happening inside the cell. Okay, both the questions are clear. Why specific growth rate is 0 for death phase, and why
Yes.
lag phase is having no increase in cell number. Okay, so the last question, question number 23. Average size of cells during exponential phase of growth is option A greater than lag phase, option B lesser than lag phase, option C equal to lag phase, and option D constant. Average size of cells during exponential phase is lesser than lag phase. Yes, option B is the correct answer. Why lesser than lag phase? So I have explained right now, in lag phase there is an increase in mass because all these synthesis of enzymes and everything is taking place inside the cell. But in exponential phase, or in the log phase as you can say, bacteria divides continuously at a constant rate and the number of bacteria increases exponentially. So in this phase, all bacteria are in their rapid stage of cell division and show balanced growth. So due to rapid cell division, bacteria have the smallest size in this phase. Okay, so they are continuously dividing. So one cell became two, then immediately those two cells enter in the division cycle and they start dividing into two more cells. So like that. So each cell is continuously dividing. So they don't have the time where they are sitting and increasing in size. Okay, so they are smallest in size in the log phase. This is clear? So this was the last question of this session. So any doubt in any of the questions, or if you want me to show any
Ma'am,
of the
ninth
questions?
question, can you please repeat?
Question number 9.
Yes, ma'am.
Second. Water activity. So what do you like, you did not understand the term, or do you have any
Yes,
specific
ma'am.
doubt?
Just you can explain that question. Actually, on that time, my network is very poor, and because of
Oh,
that,
okay.
I left the class.
Okay. So the question was, which of the following terms is like definition is given and we have to tell which of the following term is uh, defined by this definition. So basically, water activity is the ratio between the partial vapor pressure of the food when in completely undisturbed balance in the surrounding air medium and the vapor pressure of the distilled water under identical conditions. A water activity of 0.8 means the vapor pressure is 80% of pure water. So what happens is, water activity is basically the ratio of the vapor pressure of any food item, and it's basically the ratio of the vapor pressure of any food item versus the vapor pressure of the distilled water or the pure water. So distilled water or pure water, it does not contain any ions. Okay, so the ionic strength of the pure water is almost equal to 0. The conductivity of pure water is almost equal to 0. So what happens is, any so that's why the water activity is defined in terms of pure water. So ionic strength of uh, pure water is 0, so the water activity for pure water will be 1, and depending on the water activity of pure water, we will define the water activity of other food items based on the ionic strength of those food items. Okay, so let's say any food item, it will have some or the other amount of ionic strength, like it will be definitely greater than water, right? It will have some or the other ions. So what happens, the water activity will be lesser than the pure water. So for pure water, the water activity is 1, since it's the ratio of the vapor pressure, so ratio of something to itself will be 1. So water activity of pure water is 1, and water activity of any other food item, it is the ratio of the vapor pressure of that food item with respect to water. So it will be something lesser than 1. So let's say water activity of any food item is 0.8. That means vapor pressure is 80% of the pure water. So if the vapor pressure of pure water is 100, the vapor pressure of that food item is 80. And this term is actually used to define whether a microorganism can, like how well a microorganism can grow in that particular food item, using that food item. So if high ionic strength is present, that means the water activity will be very low. Okay, so there will be very few types of microorganisms that can survive in such high ionic strength solutions. Okay, if the ionic strength is very low, almost near to 0, then also since there are very few ions present, it will be difficult for a bacteria to survive. So water activity very close to pure water, and water activity uh, very less than pure water, both will be actually difficult for certain bacteria to grow. But then there are certain bacteria that can grow even in pure water, or there are certain bacteria that can grow in the food items that have a water activity that is very less than 1. Okay, so those are some kinds of extremophiles, but then most of the uh, microorganisms, they grow in the food items which have water activity somewhere near 0.8 to 0.9, which is actually like optimum for the microorganisms. Is that clear?
Clear.
Any other question?
Ma'am, yes, ma'am, not from this subject, not from this unit. Actually, I saw some questions from first unit law. Is there the question number, ma'am?
Sorry.
1 minute. Ma'am, yes, the question number 17 in the when you teach now on the time of your unit 1, log, chapter 1, log,
uh,
first unit acid base and that.
first session, right? Yes.
Yes, ma'am. In that, ma'am, the question number 17, I'll try to calculate it, but I'm not able to calculate it. Like, if it is possible in the next session or like whenever you can
uh,
read, let me.
yeah,
The
I
question is, ma'am, the buffer solutions contain 450 mg per liter acetic acid
Yeah,
and
I'm
150.
opening the question, just a second. I
Yes,
won't be able to share that question right now. It's on a different system, but I can see the question, and you have the question in front of you, right?
yes, ma'am.
Yeah, let me just question number 17 you said, right? A buffer solution contains 450 mg per liter of acetic acid and 150 mg per liter of sodium acetate. This only, right?
Yes,
Yeah. So a buffer solution contains 450 mg per liter of acetic acid and 150 mg per liter of sodium acetate. Approximate pH of the solution after addition of 15 mg per liter of HCl is. So pK is given for acetic acid, that is 4.74, right? So you have the concentration of acetic acid and sodium acetate in mg per liter. So first we can convert it into molar. Okay, so is in this part, do you have any doubt how to convert from mg per liter to molar?
No, ma'am. You can explain, ma'am, once later on.
Yeah, so 450 mg per liter, that means 450 mg is present in 1 liter. And what is the definition of molar? Molar means grams per moles per liter, number of moles per liter. So first we'll have to find the number of moles. So basically 450 mg means 450 divided by 1000, that is 450, 450 divided by 1000 will be converting into grams, and then grams, that much amount of grams divided by the number of divided by the molar mass of the acetic acid. So molar mass of acetic acid, molecular weight of acetic acid is 60, right? So 450 divided by 1000 divided by 60. So 450 divided by 1000 is basically 0.45. So 0.45 grams divided by 60 grams will be the number of moles. Okay, and that number of moles per liter, since in the question it is in per liter only, so that will be the molar molarity of acetic acid, right? This is clear?
Yes, ma'am.
Okay, so like that we have calculated the molarity of acetic acid to be 0.0075, and molarity of sodium acetate. Sodium acetate also we did the same thing. We divided 150 by 1000, that is 0.15. Then 0.15 divided by 82. 82 is the molecular weight of sodium acetate. Okay, and the answer we get is 0.0018 molar. Okay,
Okay, ma'am.
this much is clear.
Clear.
Yeah. So after that, now what is in the question? What's the solution? Total molarity, approximate pH. So we have to find the pH of the solution, right? So pH, for calculating the pH of the solution, we have to see how much salt is present and how much acid is present, right? Using the Henderson equation, we will calculate.
Yes, ma'am.
So for that, you have the acetic acid. So acetic acid will dissociate into acetate ion, CH3CO minus plus H plus, right?
Hmm. Yes, ma'am.
This is clear.
Clear.
So the concentration of HCl, so in the question you have, so after addition of 15 mg per liter of HCl, what will be the pH they have asked, right? So again you will calculate the concentration of HCl in terms of molarity. So 15 mg per liter means 15 divided by 1000, that is 0.015, and then that divided by 36.5, that is the molecular weight of HCl. So 0.0004 molar HCl concentration is there. This is also clear. Then so 0.0004 molar HCl basically will neutralize the sodium acetate. So the concentration of sodium acetate will decrease by that amount, and so concentration of acetic acid will increase by that amount. You are adding acid, right? So the concentration of acid will increase. Okay, the concentration of acetic acid was 0.0075 molar. So you will add 0.0004 molar plus 0.0075 molar. Okay, that will be the final concentration of acid, and final concentration of salt will be whatever the concentration of sodium acetate was there minus the concentration of HCl. This is clear?
Yes, ma'am.
Yeah, so then you just have to substitute in the formula. So Henderson equation is pH is equal to pKa plus log salt concentration upon acid concentration. So salt concentration is 0.0014, acid concentration is 0.0079. Clear?
Yes, ma'am.
Then you can just solve the equation to get the answer.
Ma'am, my second question is, ma'am, like when we are using that big numbers, like for example, anything minus log divided by 2 into 2.4 into 10 to the power 3,
Yes.
then suppose we are go and like I'll just check with the Google which kind of calculator used by NPTEL and all. So, ma'am, there is a very not options like in this normal calculator, whatever I'll use now, is there I can split put the value all full value like log and all big value they can give the answer, but then that have to be solved. The similar kind of calculator only we get. If you want, then I'll show. I have opened this calculator.
Yeah. You can, like, I did not get your question. What is
Ma'am,
it?
my question is that, like in the final exam, when we are go for the exam, like which kind of calculator we are just like computer based calculator or
Computer based calculators only you will get in the online exam, but in that you will get the option of solving logs, so you might have to solve it in parts. So if you have a whole equation inside the log brackets, so first try to solve the equation. So if it is log 2.3 minus 3 point something minus something plus something like that, so first try to solve that part and then take the log of the final number. Understood?
fine. Understood.
So if it is log a minus b minus a minus b plus c plus d, so first solve a minus b plus c plus d, and then whatever final answer you are getting x, just take log of that x. Okay?
Thank you.
I hope that
That's
is
it.
clear. Any other question?
No, ma'am. Thank you.
Yeah, okay. So thank you. Thank you for attending the